Okay, well, good afternoon, everybody, and welcome back to the Harmful Algal Bloom Research Symposium. The Algal Bloom Action Team is thrilled to be hosting this event for the third year, and we're glad that you can join us. This year's symposium is organized into 4 sessions over 2 days. Hopefully you joined us this morning for the first session, and this afternoon we're going to focus on harmful algal bloom detection and treatment. Tomorrow's sessions will focus on ecology and human health and case studies of effective and innovative bloom management. My name is Laura Essman, and I'm from the Indiana Water Resources Research Center at Purdue University, and I will be your moderator for this afternoon's session. Each session will be facilitated by a member of the Algal Bloom Action Team, and we are a collaboration of water professionals, researchers, and educators from the 12 states in the North Central region of the United States. We include university partners and extension, and all within the North Central region. In addition to hosting this annual symposium, our team is developing a website of resources that can be used freely for outreach and education around harmful algal blooms in the North Central region. I encourage each of you to visit our website, and hopefully Anne can post that in our chat here for you. Otherwise, I'll get it after I'm done speaking. And at our website, we are you can explore some of the resources we have to date, which include a what you should know fact sheet, a fact sheet on preventing and treating HABs for landowners, and a HABs frequently asked questions database. In addition to the symposium, we also host a bi-monthly webinar series where we feature the latest HAB research all year round. On our website, you can find recordings of all of our past webinars. You can find information on upcoming webinars, as well as access information and recordings on our past HAB research symposiums. Before we get started, I would like to just do go through a few housekeeping items. We have 3 20-minute presentations, each followed by a 10-minute Q&A session. There will be a break after presentation number 2, and we'll come back for the final one for the day. Again, we will allow time for Q&A after each presentation. To have your questions addressed during this time, please post them using the Q&A panel, which is located at the bottom of your Zoom screen. If there's a question already posted to the Q&A panel and you have the same question, then please use the Zoom upvote function on the similar question to help ensure we get answers to the most popular questions during the panel. Well, we're not doing a panel, but during the Q&A session. Please note that the Q&A panel function is separate from the chat function in Zoom. Again, questions for the presenters should be posted in the Q&A panel, but if you're having technical difficulties during the symposium, please let us know using the chat function, and we will be happy to assist you as best we can. You can also use the chat function to introduce yourself and discuss the research findings with fellow water professionals at the symposium today. Please note that all symposium presentations are being recorded and will be posted to the Algal Bloom Action Team website at the conclusion of this event tomorrow. We will also post the presentation slides to this website for your reference. And finally, before we get started, we would like to thank our symposium sponsors. We would like to give a special thanks to our supporter sponsors, which is Eutrophics and Limnotech, and for our partner sponsor, FlowCam, for making this event possible today. Okay, are we ready with our first speaker? We were having some technical difficulties.
Wes, are you able to hear us and unmute? Looks like his colleague Scott might have to pinch hit. I do see a
all right.
Wes, but
yes, I'm available. West is having some kind of a technical issue, and
can you hear me?
Oh, I hear West.
Okay, I'm on the phone, but I think I'm trying to get the computer to behave, but I think, Scott, if you don't mind getting started, that'd be great. I'll see if I
Okay.
can jump back in.
Am I sharing the slides from my end, Ann, or?
I'm going to stop sharing and I'm going to let you introduce yourself, Scott.
Okay, okay. Well, I'm Scott Schuler, the National Manager of Utrifix. And as of about 4 minutes ago, I'm going to give a presentation that Dr. Wes Bishop has prepared on preventative and early intervention in harmful algal blooms. So preventative management and early intervention. So I will do my best to get started, and hopefully Wes can get going with his technology. All right.
Scott, I can share the slides if you'd like.
I should be able to share them here in just a second. All right. Is that working?
Looks great.
All right. Are you seeing the slide? Sorry.
We
Yes.
are. It looks great.
Okay. All right. Well, as I mentioned, Dr. Wes Bishop, he's our algal and water quality research manager at our research facility in North Carolina. And I manage our Utrifix division, which some of the projects that will be highlighted in this presentation are projects that we're working on at Utrifix. Our focus at Utrifix is primarily nutrient mitigation in freshwater systems, but also we do get involved in some large-scale HAB management projects as well. So I'll talk a little bit about that. We all know harmful algal blooms can be a significant problem, and it looks like my colleague, Dr. Wes Bishop, has just joined us, so perhaps he can take over. Maybe. So we work to come up with proactive treatment programs, whether they be using algicides, copper-based algicides, peroxide-based algicides, or we work to restore water quality, which leads to a lower trophic state or a lower level of eutrophication, and tend to have less harmful algal blooms when you have better water quality. I'll talk a little bit about what we call the action threshold response or approach. This is a very proactive way to treat blooms with algicides. You utilize a lot of monitoring data, and you develop thresholds where you can prevent those blooms from expanding. And it varies from year to year based on weather primarily. Or weather can be a big driver sometimes. Some years we can get by with a single treatment. Other years we might need 4 or 5 treatments. So a lot of different factors can affect that action threshold approach. But by setting those thresholds, we can generally stay ahead of a bloom.
Great job, Scott. I'm on. Can you hear me?
I can advance those slides for you now.
Please do. I don't want to mess it up. All right. Thank you everyone for your patience and time. You think I'd be better at this by now, but appreciate that. It is very much a team effort, so I want to appreciate my team members for helping me out there. Thank you, Scott. I do want to go through a number of things, as Scott mentioned, a number of different programs we've done to both control and mitigate HABs. This is a very much an applied management talk. I am going to move pretty quickly through some of these, especially now, but we do have links to full case studies and publications for some of them. We can provide a lot more detail later, but I'm going to give a pretty quick overview. But one slide overview on harmful algal blooms here. I think you guys know what they are from many past sessions and why you're on today, but they do seem to be getting worse. We're seeing more and more harmful algal blooms in many different systems and types of systems, even in cooler water systems, the duration of blooms, new species arising. So we do, with a lot of unique environmental conditions occurring, do tend to see more harmful algal blooms, these being cyanobacteria in particular. So going to the next slide, one of the key things we've looked at as a proactive mitigation technique. Hey, how can we offset these blooms from occurring or getting as bad? One of the things we focused on is phosphorus. And I don't want to get into a big debate on, hey, nitrogen's important and the ratios are important. All that's very true. There's a lot of other things that can impact HABs, temperature, mixing, so forth, but phosphorus is one that does seem to highly correlate to both the presence and dominance of cyanobacteria harmful algal blooms in many scenarios. It's not perfect. There's always exceptions to the rule, which we can talk about, but phosphorus in particular and activation or interception is the focus of our proactive mitigation efforts for this presentation. So one of the main tools toward the next slide that we use for phosphorus mitigation is called lanthanum modified bentonite. That'll be discussed on the next slide, but here's just an overview of the intensity of the phosphorus issue across the country, and just the massive amount of pounds leading into our water resources. So the tool we're mainly utilizing in our management programs here again is this lanthanum modified bentonite. Many reasons for this. This is considered a rare earth element, but it's not that rare. It's very common in the Earth's crust. It's just sort of a title given to it, but it has a very high affinity toward phosphorus, a very stable mineral formed in the sediments that doesn't re-release under any normal environmental conditions. So again, we've done a lot of work with this over the past decade, I would say, really increased adoption here in the United States and seen some very neat results with using this technology to bind phosphorus in surface water very efficiently with a very good ecotox profile. So just to jump through some case studies here, the first one I want to talk about, going to the next slide, would be
Sorry.
oh, you're good. Would be Kitsap Lake in the state of Washington. Not a huge watershed. We found a lot of the main source of nutrients here and phosphorus was internal. So that's where something like a lanthanum modified bentonite excels. The bottom water goes anoxic, as many types of systems do. Cyanobacteria common in late summer and fall. Consistently closing down this system. So we went in, we calculated a number of different things in terms of amount of phosphorus that's being released, phosphorus fractionation in the sediments, and what's going anaerobic down there, those depth contours to target with this lanthanum modified bentonite application. So going into the next slide, you can see just what this looks like. This is a little video, but this is the application of that lanthanum modified bentonite product. It is lanthanum again on a bentonite clay carrier, and that ultimately moves down through the water column and alters the sediment release profile of phosphorus as well. All right. So here's just some of the results. Again, this is a high level overview. We will have links to to the full case studies. We have neat summary videos here as well you can watch, but in general, we met our goals. It looked a lot better, less harmful algal blooms, but we were able to track that numerically as well with both water clarity and that release of hypoliminetic phosphorus, that deep water phosphorus graph you see there, much lower following the application, the strategic application to those deep water areas to prevent that sediment release of phosphorus. So this is an ongoing program. We have been continually doing these applications to address that mobile phosphorus we've measured, but it has been very successful here in the state of Washington and Kitsap Lake, meeting their water quality goals. All right, looking toward the next study here. This is a real neat system. Some of you may have seen the news articles in Dogstein, but this is a very unique run-of-the-river impoundment, very nutrient-rich substrates here in this system. It is down in Texas, and they have issues with mats, benthic mats of cyanobacteria that often pop up to the surface, and a whole mess of different species. But the main concern is they produce a toxin called dihydroanatoxin, and it's very potent to mammals, and it's caused many dogs to die historically. So, you know, the city here is doing a lot of different things and looking at sources and watershed, which is very important. But we also did some work on the in-lake mitigation. How do we shift what's going on in the system itself to offset these harmful algal blooms and associated toxins? And again, lanthanum modified bentonite was the tool utilized here. So looking at the next slide, we see the application again, just to a section of this, the upper section, if you will, of Lady Bird Lake, where a lot of the harmful algal blooms, these benthic mats, were being identified, and where some of the dog issues were noted. And we applied over multiple applications to keep touching up those areas. And in this 20-acre section, and saw again very positive results. That's just a picture of the application going out as well. And there's a lot more detail on the project in the Utrifix video links, and I'll tell you that website at the end of the talk too. So going to the next slide, there we see, and one more click is good too, but we see the shift in mobile phosphorus fractions in the sediment. So that's what the lanthanum is designed to do, is bind that free phosphorus, the mobile, the reductant soluble forms, and shift those to that mineral form, which would come out in that apatite residual fraction. So we're seeing this big shift on forms of phosphorus in the sediment that these cyanobacteria could access, could utilize, and a shift to essentially a tightly bound mineral form that's not easily accessible or readily available for use. So that's important to note, and certainly critical in the cyanobacteria ecology, because they cause things to go anaerobic and low redox under those mats. They want those nutrients to come out of the sediments, but we're able to shift that to forms that aren't redox sensitive. So positive results there. We also saw on the next slide, we saw some things, very, very neat shifts visually. We got some samples. It's hard to quantify sometimes, periphyton and counts per meter squared, but visually we did see shifts from those cyanobacterium mats to more diatoms, to even charophytes we saw growing following this inactivation of phosphorus, and ultimately meeting objectives on keeping the system open. A lot of recreational activities go on here, and not having any dog deaths following the application. So this is ongoing work. There's certainly other areas we intend to target and look at, upstream input areas, but very positive results in this, section of Lady Bird Lake in Texas at shifting away from these toxic cyanobacteria. Another case study here. This one's in Michigan, a very neat site, about 330 acres, and we just targeted that deep contour again. So sometimes we see these hotspot areas, I call them this depth contour, where things go anoxic, anaerobic, as being large contributors of that internal phosphorus loading. So again, we're able to target that with lanthanum modified bentonite in this lake in Michigan. We are in the 3-year program as well. I'll show you the results. We've been doing small applications, I'll say, each year for 3 years, building up toward that complete inactivation of the organic and labile and reductant soluble, those forms of phosphorus able to be released out of the sediment. So quick graph there shows that the total phosphorus concentrations in the bottom water, we did see good decreases. There's still plenty of phosphorus there, but good decreases in that, and large decreases in the harmful algal blooms following an activation of the phosphorus. The next slide shows alteration in those phosphorus fractions. Again, a decrease in the labile and reductant soluble phosphorus forms in the sediment that could be released. So we have been taking multiple sediment samples from different areas and tracking those fractions through time, innate variability in the sediments, but pretty consistent decreases over this program, this multi-year treatment program, in how much phosphorus is in those releasable fractions, and also how much actually came out of the sediments to the water to support those blooms. So good impacts on limiting phosphorus and ultimately limiting the harmful algal blooms here in Morrison Lake. All right, continuing on, switching gears a little bit. So this section now is the action threshold section. So this is, a little bit different. That first section was just on, hey, what can we do, completely proactive binding phosphorus, nutrient mitigation, and offsetting algal blooms. This section's a little different. This section acknowledges, hey, sometimes those cyanobacteria are tricky. Sometimes you can't pinpoint all those sources of nutrients. Sometimes the cyanobacteria can access them from different areas, or there's inputs that are hard to pinpoint and offset. So this section is very much on, hey, how do we preserve a system and restore water resource uses if you're having blooms, or if you've had blooms that are impacting those designated uses. Now, one of the first examples I'll go more extensively through is on drinking water. So these are all just articles across the country on how cyanobacteria harmful algal blooms have impacted source water, drinking water. Many of you have heard about Toledo and so forth, but there's many other plants and issues where, cyanobacteria have caused quite large concerns in potable water sources. So looking at the next slide here, when cyanobacteria are in that surface water, that source water can cause many different issues. I know we have some supply managers on the call today, but many different issues. Taste and odor is a big one, but certainly more attention to toxins, especially in some states, and certainly concerns with human health. So, our focus has been to, hey, try to mitigate, try to offset that bloom out in the source water so it never comes into the plant. So you don't have to worry about, hey, how efficient you are at removing it or oxidizing it, or what, disinfection byproducts could be formed by sticking, chlorine on some of those toxins, for example. It's pretty scary. So trying to mitigate it in the source water and keep that water coming into the plant cleaner, and less you have to do with it to make sure it's in good shape going out. All right, so looking at the next slide, this action threshold approach, I often try to compare it to something like mowing your grass. If it gets too high, too much out of line, you mow it, you bring it back down. Is we set these action threshold levels to preserve those management objectives. And these can be adaptable. They can be, with different types of systems. They may be higher or lower than others, or if the plant can mitigate certain levels of toxins, we may increase or decrease those levels. But again, it's meant to be an adaptable program to preserve those objectives, set those conservative thresholds, and never let those cyanobacteria get above levels that could cause concern, that could produce enough toxin or taste and odor compounds to impact drinking water integrity, for example. So that's the basis of the approach, and we've applied it to many different markets, many different types of water systems. And again, it's meant to be adaptable. All right, looking at some of these triggers for potable water in particular. Again, taste and odor is a big one, and certainly associated with cyanobacteria. It could be associated with many other types of algae, as you see there, not just cyanobacteria, but taste and odor can be a big one. Certainly toxins, filter runtime, pH, just the plant functioning is an aspect as well. If you have too much cells or too much biomass coming in, it's a mess trying to get that clean and get it through their filter systems. So there's many different action threshold levels that could incite or instigate a treatment to keep that water clean. These are just a few more specifically toward drinking water that we utilize. All right, so just a couple quick examples I'll run through here. This is one. We had set our cyanobacteria action threshold in this potable source water at 5,000 cells per mil. And again, we did that. It's very low level, but we did that to ensure that, again, very minimal taste and odor and toxins have been documented at those low densities and vast majority of the time. We did add a separate action threshold level for dinobrion, some of these chrysophytes. This one in particular had a very fishy smell even at those low densities as well. So we had a separate threshold level that we added for that different algae, not a cyanobacterium. So I'm going to roll through some graphs here, a number of results. These are, through time, many different years. On this next slide, if it advances, those red arrows represent an algicide treatment. This is a copper-based algicide and water quality enhancer. So this is back when we started the program, we were monitoring those densities at 3 different depths, the drinking water plant was. And if we exceeded that 5,000 trigger, we implemented a treatment. You see it drop it down. It often comes back up. And we did that treatment again. So very, very effective results. Note some years it starts earlier than others. Note, we had late blooms. We had blooms even late into September. in that year, and it was just at depth as well. Sometimes they sit right on the thermocline. So different species, but very effective program. We would inject the algicide down if we saw more at different depths. The next slide is correlative taste and odor compounds with those same treatments. One more click should bring in those treatments as well, but we're able to bring down those taste and odor levels below that human detection threshold. This is for geosmin in particular, and it was, we can detect those at very low levels, 10 nanograms per liter. All right, keep moving through these pretty rapidly here. This is just another year again. The bloom got started very early. Aphanizomenon started early, but we're able to catch up with these treatments and get it back under control. Another slide here. Scott, again, very effective. A few more treatments this year and blooms different times, different species. But once we saw that creep up, we did an application and got it back under control, able to preserve the integrity of that source water. All right, keep moving here. Again, they started doing composite samples in 2016, but you see 4 applications we had to do in 2016 to keep the potable water clean. I think I have a couple more graphs that look very similar. 2019, again, very similar, required a few more applications that year. Was a warmer year, a lot of cyanobacteria, but able to get it under control. And I think I just have one more summer year here as well. So again, varies by year based on temperature, inflows. These are reservoirs, so they have a lot of nutrients and cyanobacteria that flow in and to start to grow, but able to get consistently get control in these dynamic systems, keep it under control. All right, Scott, we'll move pretty quick here to stay
West,
on
if you can
time.
wrap it up in about 1 minute, that we've got lots of questions coming in.
Yeah. Oh, perfect. All right. So here's just picture pre and post, looking good. All right, Scott, go ahead to the next. We'll go through here. Those are just some marina applications we did too. So in large lake systems, there's one Lake Okeechobee. One more quick one, Scott. Go down the next couple slides. Yeah, that was Lake Okeechobee Marina. I think there's one more, Scott. Zoom quickly through there. Keep clicking through there, Scott. We'll get to the end here. And this is Utah Lake. We're able to do some good stuff in marinas. We can skip that slide for now. But again, we were able to, even in these large multi-use reservoirs, it may not be feasible to treat the entire thing. We were able to identify these prioritized high-use zones, which could be a marina or a discharge or a park, and able to keep those clean, or it could be an area around an intake. Don't intend to treat the entirety of Lake Erie, but we could certainly keep an area around the intake much cleaner with different technology. So I'll wrap it up here, last slide, and get to some questions. But proactive management's ideal. Guys want to be proactive. Nutrient mitigation, we've done some neat things, but sometimes it's tricky. Sometimes it's too little, too late, or long term, or too expensive. So we have this action threshold in place so we can keep those areas clean, even this year, keep them usable, and offset human and animal health risks with that. So I'll guess go to the close slide here. I'll wrap it up, guys. I apologize for the technology issues and the time here. I want to thank my colleague, but I think I have just a couple minutes for questions. I ran through that pretty quick, but we can certainly follow up with links, and on the next slide, my contact information should be there to reach out directly as well. So
super. Thank you for going through that quickly, and we'll provide your contact information at the end of this session too. The first question that came in was, you suggested total phosphorus of less than 30 milligrams per liter to minimize cyanobacteria dominance risk. Is that a year-round average or a seasonal average?
Yeah, so that 30 micrograms per liter, that was sort of from publications, from published literature. You see that as, hey, that 2030 is in that, it's getting into that eutrophic range, but usually you don't have those massive blooms yet at that level. But sometimes those water levels are tricky. If you still have a big plume of phosphorus, that may come in, or maybe benthic. But that would be, that would be a sort of a one-time thing. You can't really use an average because, hey, if you have none in the spring and a big pulse in the fall, so averages are tricky to do through time. But that would be more of a, at any time of year, it'd be good to keep below those concentrations. So,
thank you. Where do you get the LMB?
So we, CPRO does offer, as a company, we offer many different products for control of nutrients as well as algae. So reach out to us directly. Certainly want to keep this as a non-commercial talk, if that makes sense, but you can contact us and we can provide information on where to get it. So
Great, thank you. A good question. Did you have to limit or stop any motorboat activity after these treatments were applied?
no, it's good practice to, sort of keep people away, but there's no, with the algicides, there's no swimming or irrigation restrictions associated with it. We don't want people too close to the application equipment, but they're very benign products in terms of human health. So there's no set regulations on setback distances or, swimming restrictions, irrigation, things like that, or, potable water use. The products used in the potable water have what's called the NSF Standard 60 certification for use in potable water, as well as EPA approval. So, we try to keep people away from just the general application staging area just for safety and not to contact the concentrate product. You have to have the right PPE for that. But in general, it's very benign once it's in the water. Did that answer the question?
Yeah, I think so.
Okay.
I think maybe they were also concerned about maybe mixing by motorboat activity, but if you just ask people to stay away, I'm sure that addresses it.
Yeah, and again, that lanthanum modified bentonite, it will settle pretty rapidly. Usually in a matter of minutes, you see it settle through the water column. But if you kept it stirred up, it would just have more contact with phosphorus in the water column. It's usually not a bad thing to have additional mixing to disperse it and get contact with the phosphates in the water. So.
Okay. Is there any way to decrease the organic bound phosphorus? In the graph, it didn't seem to shift with the application of LMB.
Yeah, the organic bound is really tricky. Recently, we've looked at there's more labile organic matter, more tightly bound forms. Some of them come out quicker. Some of them take a while to degrade based on bacteria and temperature, dissolved oxygen. So the organic fraction in and of itself could be fractioned, but we have seen in other sites, we can provide some information, a shift in sort of the labile organic matter fractions of phosphorus within a couple months following a lanthanum modified bentonite application. So there are some publications we can provide on that, but certainly some of the more tightly bound organic matter forms may take longer to ultimately be re-released. It's not all one big pulse, unfortunately. So
awesome. Let's see, one more question. There are a couple questions about costs and how much is needed, but that's very lake dependent and size dependent. So those can be answered on a personal level.
correct.
Does LMB affect phosphorus reserves stored by cyanobacteria?
So if they're stored within the cell itself, there's a lot of phosphorus in different compounds, certainly polyphosphate bodies they could store it as. If it's within the cell itself, it's not necessarily going to grab that and bind with it. It prefers those polyphosphates or free reactive phosphorus forms that are often called in the water column. So if you have an existing bloom and you use lanthanum modified bentonite on existing bloom, it's not going to suck the phosphorus out of their biomass. Oftentimes we would suggest trying to be proactive, try to do it before they get there, or utilize an algicide. We have peroxide-based algicides, for example, that could release that phosphorus to phosphate and then follow that up with the lanthanum modified bentonite. Shock and lock, I affectionately call it. But good question. Good question. So.
Well, there are some more, and maybe you can address them while the other speakers are going, or deal with, or people can contact you directly. Yep. Thank you so much for bouncing in and doing that quickly. We appreciate that. Thanks, West.
Thank you guys for your time. So
sure. Okay, I'm going to go ahead and share my screen and introduce our next speaker, who is Polly Berriman. And Polly is the Water Markets Manager at Yokogawa Fluid Imaging Technologies. I practiced that a lot, but then I messed it up anyway. She does work closely with freshwater researchers and drinking water utilities worldwide, and she's helping them to implement this FlowCam technology that she's going to tell us about very soon here. Go ahead and share your screen, Polly.
Okay, 1 second. Sorry, is that I have a bunch of screens showing up right now.
Yeah, we're seeing your presenter view.
Strange. Yeah, I've got something blocking me and I can't remove it.
Can you stop sharing and
It won't let me. Sorry, I'm gonna stop.
this always works perfectly when you test it.
I know it was great.
There you go. There you go. There you go.
Okay, we good?
You're
Okay.
good. Go ahead and take
Excellent.
the show.
All right, well, thank you for that introduction and thanks everyone for joining me today. So I'm Polly and I and my co-author and colleague Savannah are members of the aquatic science team at Yokogawa Fluid Imaging Technologies. And our company manufactures a flow imaging microscope called FlowCam. So flow imaging microscopy as a technique has been around for a couple of decades now. It was initially developed to image and count marine phytoplankton, but it's since evolved into a technology that can be used wherever particle concentration and morphology are needed. So today with this presentation, I really wanted to address the unique challenges that are presented by colonial phytoplankton when using this flow imaging microscopy technology. And I'm going to use Microcystis as a case study. So I want to start out with just a brief overview of why we chose to feature Microcystis, followed by just a general overview of how we would quantify colonial organisms like this, what techniques are used, and then jump into a little bit of the flow imaging microscopy technology, what it entails and how it relates to the quantification of colonial organisms. And then we'll review some studies that highlight some of the challenges of quantification and identification and the ways in which the authors used flow imaging microscopy to overcome them. So why Microcystis? I think usually when we think about HAB management and harmful algal blooms in general, Microcystis is one of the big ones that really sort of jumps to mind. It's been found globally, I think everywhere except for Antarctica at this point. And so it's just one of the big ones that we most often associate with HABs. And then of course, many of the species are toxin producers. And as we learned earlier, they do pose a risk to human and animal health. So it's really essential that we're able to accurately monitor these blooms and create these threshold levels for treatment. So of course, one of the real reasons that we chose Microcystis for this presentation, though, is because of the challenges that it presents with respect to quantification. For something that's relatively common and capable of producing toxins, quantifying Microcystis really presents a unique challenge, regardless of the technique that you're using. So it's important to remember that these organisms, they're a nuisance today because they've had a couple of billion years to evolve. They have incredible strategies that allow them to survive under harsh conditions and outcompete other members of the microbial community. So they're pretty cool. And they're really only problematic now because they're just really good at what they do. So when we think about quantifying Microcystis, there are various ways that we can do it. The most convenient technique, of course, involves using pigments like chlorophyll A and phycocyanin. And these pigments give you sort of a really quick overall read of what's going on. They'll give you the overall phytoplankton biomass and what proportion of it is cyanobacteria, but it doesn't tell you who's in there. And when you consider the fact that different organisms are producing different toxins, and they respond differently to certain treatments, and the fact that we also need to be able to create these threshold and treatment levels, knowing which organisms are dominant is really important. So next, biovolume is another way of quantifying cyanobacteria and algae, and it's usually calculated based on a formula that best aligns with an organism's geometry. Microcystis, though, is challenging because the colonies are variable in size and shape and structure. So it's difficult to reliably apply a one-size-fits-all approach to its geometry. So that leaves us with cell density, and this is the most common way I think of quantifying microcystis. So this is the number of cells that make up a colonial unit, and it's reported as cells per mil. So you'll recall in the previous presentation, this was how they determined, their thresholds, the cells per mil. So the challenge, though, is that counting these individual cells in a microscope is really time consuming. And due to the layers that you can see here that make up the colony, the numbers reported are usually kind of ultimately an estimate. So just to put that into perspective, here's a small microcystis colony, and you can see here that the individual cells are really easy to count. You can see, I think there's 1, 2, 3, 4, 5, 6, 7 colonies in this. But now if I move on to the next slide, you can see looking at these colonies, it's really easy to see that it would take a really long time to count all of the cells. And even then it's going to be a challenge for some of these colonies where the cells are really close together and overlapping. So ultimately counting individual cells is time consuming. Measuring cells to calculate the biovolume, that's also going to add a lot more time. And really, no matter how you do it, there are calculations and assumptions involved. So at the end of the day, automated imaging techniques can save a lot of time, but there are some key considerations that we'll move on to discuss. But first, just to give a little bit of background on flow imaging microscopy as a technique, it combines the visual element of microscopy with imaging in flow. So this means that when the sample is introduced to the sample port in the top, it's pulled through a flow cell by a syringe pump. And in the flow cell, it's kind of like a photo booth. The particle gets its picture taken. The camera flashes and the software automatically separates the individual particles and saves the images in the software along with a number of different particle properties. And techniques like flow imaging microscopy that help to speed up this data acquisition have some benefits and limitations, as with all technology. On the benefit side, you can automatically acquire thousands of images in a really short period of time. And these are automatically saved to the database. You can pull them up whenever you need to. You're also able to record a number of different measurements, such as diameter, area, biovolume, and a number of others as well. But colonial organisms like microcystis do present some challenges when using flow imaging microscopy. So the first is, images are captured as just that, an image. So as it flows through, the image is counted, but it's counted as one natural unit. So additional steps are required to get an estimate of the cell count for these colonial organisms. It's also important to consider that it records a two-dimensional picture, and this can't be manipulated. So as we saw in previous slides, the cells are likely to overlap. This can make it really difficult to estimate the number of cells in that colony. Also, depending on the sample type, size fractionation might be necessary to help prevent clogging of that flow cell. That's pretty easy to do, though, not very time consuming. And then lastly, image recognition using libraries can be difficult for colonies that vary significantly in size, shape, or distance between the cells, as with microcystis. Over the past decade, though, there's been some really interesting research into how to overcome some of these challenges. And so next, I want to go into some of these studies and discuss what was done, how it was accomplished, and some of the key takeaways that we could apply to future work. So important to note, first of all, in South Korea, microcystis is regulated based on the total number of cells within a colony. So researchers who are looking up to speed up this analysis by using flow imaging microscopy need to ensure the accuracy of their reporting results. So in this case, in this paper by Park et al., what they did was to develop a straightforward algorithm for converting 2D images, like the ones that we just saw with the flow imaging microscopy, into 3-dimensional cell counts. So as I mentioned earlier, one of the challenges with visual techniques is taking into consideration this 2-dimensional picture that you're looking at exists in 3 dimensions. And how do you account for this? So to do this, first they collected some samples from various sites along 2 local rivers in South Korea. And then they ran replicates of each sample on the FlowCam. For anyone who's participating, who's familiar with the FlowCam, they used a 4X objective with a 300 micron flow cell. This was to accommodate some of the larger colonies. And from built a library of images that had 2,520. So this was a sizable library. And then they needed a way to capture the size of each colony in 2 dimensions. So they used area. And this is one of the properties that imaging devices like this can automatically measure. And after summing the area of all the colonies, they performed manual cell counts on a microscope by sonicating the samples and adding sodium hydroxide to separate the colonies into individual cells. And then they ran a regression to associate colony area to the number of cells. And then, of course, the last step was to account for bias between flow imaging microscopy and the microscope. So what Park et al. Found was a strong correlation between colony area and the number of cells, but a lower correlation between cells from the FlowCam compared to microscopy. So the bias correction was necessary to account for that difference, and it allowed them to generate a correction factor that could be applied to future samples. So really, I think the take-home message for this study was that it's important not to forget that working with 2-dimensional images of 3-dimensional objects is going to inherently introduce a certain bias to organisms like Microcystis, where those cells may be overlapping each other. But this bias can be accounted for by combining manual microscopy and flow imaging microscopy. And hopefully this would just be for a subset of your samples. Okay, so moving on to a paper by Wang et al. And this is from 2014. So this is an oldie at this point. And the goal of this study was to develop a comprehensive method to rapidly and accurately analyze cell density and colony size distribution. There were 3 core aims in this study. The first was to optimize a technique for breaking up or disintegrating a colony into single cells for enumeration. The second was to optimize a procedure for isolating Microcystis from the sediment. And the last element was to validate the accuracy and precision of using flow imaging microscopy to enumerate the cells using their isolation procedures. And I think it's really important to note here also that they didn't neglect the benthic Microcystis. As we know, sinking down to the benthos is how Microcystis will overwinter. And so it's important to get if we want to get the full picture of what's going on, that we don't neglect this. So to accomplish this, they started with the first core element, and they sought to optimize a procedure for disintegrating Microcystis colonies for enumeration. It's a little bit of a different approach from the paper we just reviewed by Park et al., where they accounted for the bias correction. In this paper, Wang et al. Opted to separate the cells and then count them. So they compared 3 different techniques to break up the colonies in order to find the most efficient, and these included sonication, heating, and alkaline hydrolysis. Keeping in mind that different species of Microcystis may respond differently to each of these treatments, they tested 5 key species and also 1 wild sample, which contained 4 of the 5 species. And to evaluate each technique, the researchers looked at the efficiency of the disintegration and found that it varied between treatments, morphospecies, and concentrations. So it got quite complicated. The striped bars represent 3 different sonication intensities. The efficiency of disintegration was found to increase with sonication intensity for some morphospecies, like the M. viridis, but it decreased for others like M. wessenbergii. So these results indicate that even though ultrasonication is a common method for disintegrating microcystis, and it was one of the methods used by Park et al. Earlier, it's not consistently efficient. Heating at 80 degrees centigrade, and that's represented by the solid black bar, also had a high degree of variability. So if we look at this, ultimately you can see it was the alkaline hydrolysis at 0.01 moles per liter of sodium hydroxide at 80 degrees C that produced the most consistently efficient results. Using higher concentrations of sodium hydroxide was found to cause the cells to break. So moving on to the second core element of their study, and this was to optimize a method for the isolation of benthic microcystis from the sediment. And the researchers used cultures to develop the method, and then they tested it using field samples. So to prepare their culture samples, they mixed 1 gram of sediment and 1 ml of water with microcystis of a known concentration. They refrigerated it, then suspended the sample in 5 ml of Percol, mixed at various concentrations. And then they centrifuged the samples at various RPMs. And then finally, they diluted the supernatant to a standard volume and counted how many cells there were compared with how many they started with. So for their field samples, they collected sediment and water samples from 12 locations on a single lake. And of those, they pulled out the samples with the highest concentration of microcystis. Then they analyzed the sample, the sediment samples 6 times using the disintegration and isolation procedures to see how they would perform. And then lastly, the final pillar of the study was the evaluation of the accuracy and precision of FlowCam for enumerating their samples. So just like with their other methodologies, they use both cultures and wild samples. And they used the cultures to enumerate the individual cells of microcystis, since it doesn't form colonies in culture. They also wanted to evaluate the accuracy and precision of sizing colonies of microcystis. And to do this, they collected nettos and separated the colonies into 3 size classes. So the colony size based on length is measured 6 times with FlowCam and with the microscope respectively. And for counting the unicellular cultures, they found that counts were very similar. There was some degree of variation between samples, with the mean coefficient of variation landing at 4.5%. So when comparing the accuracy and precision for counting the number of colonies per mil, they found that there was no significant difference between FlowCam and microscope for small, medium, or large colonies. And really, this isn't surprising when you consider that flow imaging microscopy counts each colony as a natural unit. So that comparison is quite straightforward. The highest amount of variation between samples you can see was in the small size class. So from this study, there were a couple of key take-home messages. The authors developed a rapid and reliable method for measuring cell density and colony size distribution, both in the water column and the sediment. And through a lot of testing, they found the optimal hydrolysis and Percol procedures to disintegrate and isolate benthic colonies. And then lastly, they were able to apply these protocols to quantify microcystis in a lake environment. And of course, it was also really important to them to study both microcystis in the water column and in the sediment, because both techniques are really important if you want to investigate bloom dynamics over the course of a year. So moving into the last and final paper that I want to highlight, and this one is a little bit different. It speaks to the challenges surrounding identification. So far, we've looked at focusing on overcoming challenges with respect to enumerating colonial organisms. But I think identification is also worth touching on. So in this study, the researchers used a mesocosm to collect training images for a machine learning-based approach to identify Microcystis from 2 other genera. And then they took it a step further to develop a method for identifying 5 morphospecies within the Microcystis genus. So here are some examples of the target groups. So you can see between the genre, they sought to distinguish Microcystis from Cryptomonas. That's quite straightforward. A little bit more challenging for Microctinium. And then within the Microcystis genus, they wanted to separate 5 different morphospecies. So to do this, they sampled from different microcosms on different days and preserved them with glutaraldehyde. For the FlowChem analysis, they pre-filtered their samples to target the colonies that were under 100 microns so that they didn't have to deal with any clogging of the flow cells. Initially, they had to contend with how to handle messy colonies. These were the ones that were only the remnants of a colony that were left behind, or they were too dispersed and undefined. And then for each group, they randomly selected 150 representative images. So the FlowChem software automatically will record different particle properties. And they wanted to develop a filter set that was based on properties showing the lowest degree of deviation. So as a result, they decided to only use the particle properties that led to an accuracy of 0.75 or above. And you can see here that the performance for the intergeneric classification, the one between the 3 broader groups, was better than the intrageneric classification. The 3 broad groups were pretty unique from each other, so it was easier to achieve, I guess, a better result with this, but it was a little bit harder with the Microcystis groups, as they looked a little bit more similar.
Polly, we have about a minute.
Oh, okay. I'll go to our key takeaways and then I'm almost done. So really what you can achieve with this study, the key takeaway from this study is that you can achieve classification accuracy that's comparable to traditional methods or better, and save up to 80% of a person's time. So I think that's really important. It also highlighted the utility of microcosms for conducting studies on seasonal succession. And so really just to summarize, I think it's important to note that Microcystis, amongst others, is important for further investigation. Ultimately, digital microscopy techniques like FlowChem can save time, but you do need to understand the unique challenges and address these challenges of colonial organisms. And so these are the references that we used for this presentation. If you'd like to read those papers for yourself, I can also send this to you afterwards if needed. And this is my contact information. So hopefully we have time for questions, and I will stop sharing.
We do. Thank you very much. We've got a couple minutes here. Okay, so everybody's challenging me on my pronunciations today, but I will do my best. In the flow imaging microscope slide, which was about 11 or 12, slide 11 or 12, is the genera between the examples of Microcystis and Delictospermum and Aphanocapsa species.
So I'm looking at slide 11. So this was the one where we were just looking at some dense colonies. So I believe this was actually taken from, I'd have to double check this, but I think we took this from a paper, and these were all classified as microcystis. So that I'm not sure if that's incorrect though.
All righty. Does FlowCam identify, whoa, my questions are disappearing. Sorry, they're moving around. Does FlowCam identify and attempt to quantify strains of cyanobacteria besides microcystis?
Yes, so FlowCam, as I mentioned at the beginning, it will image and count any particles flowing through the flow cell. So from that, you can build libraries of organisms, and we have some example libraries that come with it. But then you can classify down to any different genus that you're interested in. So if you want to look at everything that's in your sample, that's completely possible to do.
Great. Can you comment on how to properly collect the water samples?
So I think that's really down to your individual, however you prefer to do it. I think most utilities that I know are just doing grab samples.
Okay. Would it be legitimate to institute a standard for microcystis based simply on counts of colonies rather than individual cells? Presumably, this could account for both toxin levels and, for example, oxygen depletion potential if carefully calibrated experimentally.
That's a great question, and I would love to know the answer. And hopefully somebody will do that research so I can answer it. But yeah, I think it's certainly something that should be investigated further.
Okay. Does the alkaline hydrolysis of the colonies affect cell turgor or biovolume? Decreased turgor, decreased cell volume, underestimate of cell concentration.
So that wasn't listed in the publication. That would be a question that would be great to reach out to the authors and ask. And I can absolutely do that after this.
Okay. And then I think you've already answered this, but I'll run it by you anyway. Is the FlowCam adapted for filamentous cells?
Yes, filamentous, colonial, again, any particles flowing through. So of course you want to make sure that the configuration is appropriate for the size of the organisms, but otherwise, yes.
Okay. We do have one comment that's, a person has requested for a consult and a quote, but hasn't received follow-up. Are you a good contact for that, or could you provide a good contact for that?
Absolutely. Yes, please reach out to me. And I'll look through these questions afterwards and see if I can find the information too.
Excellent. Well, thank you very much. You ended us perfectly today, on time.
Wonderful. Thank you.
So I am going to share my screen again. And we are now going to break for 20 minutes before our final presentation. You will note there's been a little bit of a change to our schedule today. Unfortunately, the presentation from Elizabeth Gao on light-based mitigation had to be canceled today. So we do just have one more speaker after the break. So please come back in 20 minutes and join us. And special thanks again to our sponsors, Utrifix, Lemnotech, and FlowCam. .25 where I am, 2:25 Central. So we're going to get started and finish out our day here. Before we do that, I would like to welcome our last speaker, who is Daniel Swanepoel. He's a graduate research assistant at IIHR Hydroscience and Engineering at the University of Iowa. And he's currently conducting research focusing on improving the detection and monitoring and understanding of harmful algal blooms under his advisors. He's going to talk to us this afternoon about improving the spatial and temporal monitoring of harmful algal blooms in Midwest lakes using UAV remote sensing approach. I'm going to pass things over to Daniel here. and allow you to share your screen.
Mhm. How's that look?
All you have to do is put it in presenter mode and you're perfect.
There you go.
There you go. Perfect.
Cool. Sweet. Thank you so much for that introduction. Yeah, I'm pleased to present my research project titled Improving the Spatial and Temporal Monitoring of HABs in Midwest Lakes using a UAV or Unmanned Aerial Vehicle Remote Sensing Approach. This project was conducted under the guidance of Mark Lindemann, Corey Markford, and Susan Meerdink with myself. So for a quick little outline, we're going to cover the principal research question and then go over cyanobacterial HABs and the importance of monitoring these systems, then current HAB monitoring strategies and their limitations, then an overview of remote sensing methods applied to HAB monitoring. Then we're going to look at a case study that we conducted at Lake Darling. And then we will look through the data processing workflow from what we collected there. We'll check out the initial results, and then we'll go on to the summary conclusions and talk a little bit about the future work as well. So for our principal research question is essentially, can a low-cost, near-range remote sensing tool be used to effectively monitor HABs in small inland lakes? That's basically the overarching kind of big question. So for a little quick overview, as you guys probably all already know, cyanobacterial harmful algal blooms or cyanoHABs are an increasingly pressing issue as they are becoming more frequent, intense, and long-lasting due to climate warming and anthropogenic activity. These blooms can have detrimental effects on public, environmental, and economic health, as they can release toxins and deplete oxygen levels in the water when they decompose. Therefore, it is of utmost importance to detect, monitor, and understand these HABs in order to effectively address their negative consequences. Small lakes in the Midwest, which are prone to high levels of agricultural activity and the resultant nutrient runoff from those activities, are particularly susceptible to these HABs. As such, it is critical to closely monitor these bodies of water in order to mitigate the negative effects of HABs. So for current HAB monitoring strategies and their limitations, HABs are dynamic systems that can change in all three dimensions. Wind can change them, the currents created by wind, or just if you're in a small river, or just general currents, and even light conditions can affect how these systems change over their lifetimes. Some species of cyanobacteria have little gas vesicles, and under low irradiance or low levels of light, if there's a lot of overcast, those gas vesicles can actually puff up and dilate in a way, and they'll sort of adjust their level or height or depth in the water column, depending to sort of optimize the level and how much light they're getting. So this is a 3D issue. There are several methods available for monitoring HABs, each with its own advantages and limitations. Grab samples provide toxicity information via ELISA and congener information through chromatography, but these have low temporal and spatial resolutions. Imagine taking a grab sample at a single point, as you can see by this image on the right here. This is variable in space over the surface of the water. So, and this could change in a matter of hours, definitely over days. So if you take a grab sample there and you think it's okay, there could be a mess waiting kind of deeper in the deeper waters there. So same with in situ sensors and samplers. They do offer, with in situ sensing, they offer higher temporal resolution, but they still lack that spatial information. And they also are sometimes susceptible to fouling and need frequent maintenance to make sure you're getting good data out of those sensors. Then we have satellite remote sensing. This has high spatial coverage, as you can see in this image, but low spatial resolution. So you end up with mixed pixels, especially when you're looking at the smaller inland lakes. They have been used effectively very well for large ocean systems or like very big lakes. But for the smaller Midwest inland lakes, there's some issues there. They also get affected by atmospheric interferences such as clouds, aerosols, and other atmospheric gases. So, you know, that light data that you're collecting gets skewed, and there's error associated with that, which just adds complexity. Then we have, for the next method, we have near range remote sensing. This provides high spatial and temporal resolution, but it does have limited coverage. Both aircraft and unmanned aerial vehicles, or UAVs, can be used for remote sensing. And with aircraft capable of carrying larger payloads and higher-end sensors, but at a higher cost of operation and takes longer to get those in the air, while UAVs offer lower cost and easy automation, but they do have a limited payload capacity. So for higher-end, more expensive sensors are usually heavier at this point, even though they are decreasing as technology progresses. Right now, that is a slight limitation for smaller UAV platforms. Due to the higher spatial, or the extremely high spatial resolution of these near-range UAV images and the data that they collect, multiple images are often captured over the water's surface. These images then need to be correctly positioned and stitched together to create useful spatial maps that depict the extent and the intensity of these HABs, which is essentially what we want. Now, this has been a major challenge for current photogrammic softwares due to the lack of stationary sort of ground control points that you have when multiple images are over water. There's no real way to tell if that image is in the correct place, whereas a lot of, the current software uses, that's a tree, that's a rock, and you can snap them together because they're both in different images, so you know for sure that those points lie on the same point on the Earth. So looking at these, it is clear that no single monitoring strategy can adequately capture the complex spatial and temporal and toxicological dynamics of HABs. So improving the temporal and spatial resolution in HAB monitoring is critical, and near-range remote sensing using a UAV-based platform offers potential in addressing these needs. So remote sensing for HAB monitoring. So we're looking at overwater imagery here. So first, to just give you a little background for those of you who aren't too familiar with the electromagnetic spectrum. The electromagnetic spectrum essentially refers to the range of all types of electromagnetic radiation, from the longest wavelengths, such as radio waves, to the shortest wavelengths, such as gamma rays. Remote sensing reflectance is the measurement of the amount of electromagnetic radiation that is reflected off of a surface of an object. In the context of harmful algal blooms and their monitoring, different types of algae and cyanobacteria reflect different amounts of electromagnetic radiation at different wavelengths. So by measuring the reflectance of HABs at different and various wavelengths, we can use these remote sensing techniques to identify and map their distribution on the surface of the water. If you look at the plot here on the right, or on the left, this was taken from Tomlinson et al. 2015, and shows the remote sensing reflectance of a water's surface, measured using a handheld spectrometer. The vertical colored lines indicate the bands that the satellite-based multispectral tool, MERIS, measures. So just to sort of overlay that on top of the hyperspectral data that you have collected here by this spectrometer. And just to clarify, multispectral just refers to a sensor that measures a few bands of light in discrete wavelengths, as these bars indicate. And then you have a hyperspectral sensor that usually often measures many more wavelengths of light, often at very, very narrow bandwidths. So you get very, very, very narrow little bands of different wavelengths all stacked together to get this sort of spectral shape, or this reflectance shape graph here. Then what is known as a spectral algorithm or spectral index is something that can be used to highlight various constituents at or in the water surface by calculating ratios of the remote sensing reflectance at various wavelengths. So you're looking at ratios between, let's say, the near infrared and the green, because these two, the same constituent or same object or same type of algae reflects light differently at those wavelengths, and you can actually pull those constituents out by using a spectral index. Many different indexes have been developed to sort of highlight different types of constituents, such as chlorophyll or phycocyanin, and many other things like rocks and minerals on the surface of the earth. And many of these indexes are sort of specific to those constituents, and then to different water conditions in the case of HAB monitoring, and also they are specific to the sensors, so the different bands, and they are actually, a lot of them can be quite sensitive depending on exactly what wavelengths each band uses. So if you're looking at one sensor that measures red, blue, green, if they're talking about a certain wavelength band, if you look at another sensor and they also say, well, this is red, but they've maybe shifted over, that may affect any given index by a substantial amount. So if we just click through these slides here, you can see in the top right here, you have different situations that can arise when you're trying to look at this reflectance data over water for HABs. So you have just absorbing right on the surface here. Let's say this is an algal cell. You get that. Then you have incoming solar radiation. It's going through the water, it's absorbing, and then it's getting emitted and reflected. You have absorption of water and other constituents as well. So that's going to show up in the signal here. And one of the challenges there is to try and pull out the signal that only the algae or the constituents that you are concerned with are, what signal is essentially being produced by that. Then we have another issue that arises frequently, which is sun glint or specular reflections, where essentially you get your pixels and your images just get completely saturated. You can see this in this image here, and even over here. Here you have surface plants too. So when it was bouncing directly off of there, is that toxic or dangerous algae, or is that just duckweed or some other type of photosynthetic vegetation floating on the surface? Another thing is that remote sensing cannot directly detect toxins as of right now. They detect pigments. So they'll basically go and detect chlorophyll A or phycocyanin, something that is associated with these HABs. And then obviously toxins have to be inferred based on other relationships that we'll get into later. So for the case study, we looked at Lake Darling. Excuse me. Lake Darling is an Iowa lake situated in Washington County, about an hour drive from Iowa City. Over the past several decades, Lake Darling has experienced frequent microcystis-dominated harmful algal bloom events. In 2010, the lake underwent a 4-year, $13 million restoration project to improve these conditions. Unfortunately, these efforts proved ineffective as the prevalence of toxic, low visibility water conditions continue to be documented. However, these conditions provide an ideal testing site to evaluate novel HAB monitoring strategies. So we conducted a case study here in the summer of 2021 to assess the utility of a low-cost multispectral sensor for predicting chlorophyll A concentrations in the upper water column. And then we also looked to improve, so the second objective here was to improve the current methods for geolocating, projecting, and mosaicing, or stitching together high-resolution over-water imagery, which is, as I mentioned before, been quite a major challenge in these technologies. And then thirdly, to investigate whether a relationship between chlorophyll A and microcystin, is a cyanotoxin, exists in Lake Darling during this season. So 3 layers of data were collected over Lake Darling. We went in and took 10 grab samples, and they were analyzed for microcystin and then chlorophyll A and phycocyanin. So 2 pigments and a cyanotoxin. We try to sort of map these out to sort of capture any variability that was in the lake, try to spread them out to get the full extent of the lake there. We also performed 6 UAV flights, and they collected multispectral data, and you can see that by these little white polygons here. So they were flown over to capture, the water where that grab sample was taken. So we took a grab sample and then went to shore and flew the flight over that. There was also one aircraft flight that collected hyperspectral swath data over the whole lake, over the whole length of the lake. So we have that that high level, top level data layer as well. So for the grab sample data, here we have the 10 samples and the values for microcystin in micrograms per liter. We have them for chlorophyll and phycocyanin, also in micrograms per liter. So the sample locations were navigated to in a university boat and handheld GPS. Samples were collected, placed on ice, and then sent to the Iowa State Hygienic Lab for analysis. Looking at just a little regression analysis here to see if there is any relationship between chlorophyll and microcystin. By the way, this is ELISA microcystin, which is the way it was analyzed. We see, there's some relationship there. The shaded region here indicates a 95% confidence interval. There's only 10 data points, and the low variability and range in values does make it difficult to really interpret this regression analysis here. It would be nice to have, a larger range, maybe some very low to 0 chlorophyll and microcystin values to add into this to sort of stretch it out so you can really pull out if there is any stronger relationship there. To relate this to some previous work done by Douglas Green et al. 2021, I took this figure, and what happened there, they did a study where they took beach samples collected by the DNR, and that was done once a week over the whole HAB season or summer season. So you can't really compare these, fully or properly, since these grab samples were all taken in one day during one bloom event. These ones over here were taken, over the extended period of the season. So I think there definitely is more work to be done on the interseasonal stability of such relationships. But as you can see here on the left, there has been some significant strong relationships, positive relationships between chlorophyll A and microcystin. So then for the multispectral UAV imagery. So each flight with their corresponding grab samples, time of deployment, cloud cover, wave size at the time of capture, number of single band image captures, and total number of multi 5-band or composite sort of stacked images. So you have here, let's say you have number of single band images, we have 470, but since essentially there's 5 images per capture, you only have 94 captures. So there's basically 5 layers. And this is the camera we used, and this is the drone we used. This camera, essentially here you have 5 discrete lenses that measure, a different wavelength of light or a different band of light. And so here we have the flight paths for each flight there. The drone flight paths were planned using Atlas flight with an altitude of 80 meters above the ground, a speed of 5 meters per second, and an image overlap of 75%. At the flight altitude, a ground pixel resolution of 5 by 5 centimeters is achieved. So that's very, very high spatial resolution there. So looking at the multispectral image processing workflow. So once we got all of those images and we have a bunch of essentially just 5, like band 1, band 2, band 3, band 4, band 5, just a bunch of them. We essentially went and had to convert the image pixels from raw digital numbers to reflectance, so units of reflectance. Then we had to align the 5 bands for each capture. So since for each capture, those lenses are offset, you have pixel offsets. And if you want to do ratios or band math algorithms or spectral algorithms, also known, you've got to make sure that those pixels are overlapping in the correct way, and they're essentially representing the same location on the ground. Otherwise, it's going to mess up those algorithms and or index calculations. Then the third part of the workflow is to geolocate and project the images to a surface footprint using the drone and sensor orientation. So this drone is moving around, and depending on how it's moving, that image is essentially looking at different points. And then finally, image selection and spectral analysis algorithms were applied. So for the first step here, converting pixels to reflectance with a calibration panel from the camera manufacturer. So looking, this was a Red Edge 3 multispectral camera made by MicaSense. They provided a calibration panel that is used. So right before and right after each flight, images of this panel, which right here in the middle, which this is Spectralon, the material is known as Spectralon, and it's been calibrated in lab, so it has known amounts of reflectance in each of the bands that this camera measures. So in the blue, it's 0.71 and so on. So that's the known. Then what we do is we apply a camera-specific mathematical model developed by the manufacturer, and that converts all the pixels within the panel region into units of light intensity known as radiance. And here is just the main radiometric calibration model and all of their variables. Now that we have this calibration panel in units of radiance, since we have the radiance, so what's basically outgoing or coming off of that calibration panel, and we know what the reflectance is, we can essentially calculate a radiance or pixel value to reflectance conversion factor, and you can apply that to all the images within that flight. So this calibration panel has been proven effective, and then we have validated our reflectance data against spectral libraries such as the NASA Jet Propulsion EcoStress spectral library to make sure that our data there makes sense and looks good. So a little bit deeper into the MicaSense Red Edge 3 multispectral imager, you can see here that each lens measures a specific wavelength or band, and they're offset. And here is a little image or plot showing the bands here in blue. So the band and the bandwidth. So that's essentially what you're getting. And then these lightly colored, slightly transparent colored bands show the bands of interest when you're trying to detect chlorophyll A in an object or in water. So over here, I'm going to show you what happens when you look at an image and you look at two different bands of the same image or the same capture when they're not aligned properly. So you can see that they're offset there. I can do that again. They're offset, and that's an issue because you want to make sure that your pixels are overlapped. When you have things like sun glint, if you're doing a ratio between, let's say, top layer band 1 versus band 2, if you're looking at the same point, it's not the same point, so you're going to get messed up calculations, and the performance just won't be good at all. So that was a big part in the image processing workflow. So for aligning the bands, we essentially had to align them and save them out as a composite band or GeoTIFF file image. So you can see here, before they're aligned, it looks a little bit wonky. Once you align them, it looks way better. And the way we did this was, so we chose an image for each flight, a single image that was over land, that has sufficient landmarks. And we created a transformation matrix that was then generated with OpenCV library in Python to allow the alignment of the 5 bands. So you get that transformation matrix. And then once you find that matrix, since that was taken in the same flight, you can be confident that those lenses didn't get knocked around between landings, and you can apply that matrix, that transformation, throughout all of the images and get a nice solid alignment. Because if you're looking over water, it's a little bit harder because there's way less landmarks and you can't see what pixel relates to the other band's pixel. So that's why we had to do it that way. And yeah. And the resulting stacked image then needed to be cropped to exclude the pixels that do not include a pixel in all 5 bands. So if you think about over here on the left, you have this perfect image. They have the same number of pixels in X and Y directions. Once you move them to align them, now there's overlapping sections where not all the 5 layers are overlapping. So you've got to crop out to make sure that the end image doesn't have a no data value for any of the bands. For geolocation and projection is the next step. And essentially the idea here is that you get an image that is perfect square. And this is a theoretical geometry here of the drone, but in reality, this drone is moving around like crazy. And you can describe this using 3 angles in 3 dimensions: pitch, roll, and yaw. And so we use these angles that are captured at the same time as an image is captured from an onboard IMU, which is an inertial movement unit, inertial measurement unit. And so you have those 3 angles as the image is captured. So we can actually project this image using a rotation matrix going from here to here. And you can see how that's so different. If you just plot one point and paste this image down, it doesn't align with the actual ground. So if you're looking at a point here, it's not the correct point. Once you apply this method of sort of warping and projecting these images, you can see how that alignment gets a lot better. So this becomes important when you are looking at overwater images and you have no idea if that's actually projected in the right place. And if you're looking at if it's overlapping a sample and you're trying to, how well did this algorithm or this index predict the chlorophyll concentration? Are you even looking at the same point? So that's the biggest thing here that has been a huge challenge that we're mainly working on right now.
Daniel, we have about 1 minute.
Okay, cool. So another thing here is the orientation of these drones matters because we have sun reflections and they're bouncing off the ripples. So you can look at these 2 different scenarios. 2 different drone angles give you 2 very different pixel values. So that is something that we have to account for too. And yeah, that is definitely a challenge. Once we have all of these stacked images ready and warped, we can pull them into software such as ArcGIS and choose the images that are overlapping samples. We mosaic them here. So I'll just speed up here a bit. Analyzing these images covering the grab samples in ArcGIS Pro, we mosaic them. Essentially, this is just initial results, and we've taken each pixel. We have multiple images over each grab sample, and we've just taken the pixels between all of them and just averaged them into a mean and then displayed that right here. So it's pretty crude at the moment, but this is the initial point. Then applying an algorithm called NDVI, which is the near infrared minus red. band and near infrared plus red. We can see how this index predicts chlorophyll A concentrations. And here we have, using 1 pixel, the only pixel, we have a 0.37 R-squared value or correlation coefficient. When we increase that to 3 pixels, kind of dampens out some noise. We get a little bit better relationship there. And then, so for the final slide here, summary and conclusions, a weak positive correlation between chlorophyll A and microcystin was observed in Lake Darling during the summer of 2021. However, a lack of analyte variability and small data sets reduces its certainty. The interseasonal stability of such toxin-pigment relationships should be investigated to assess the utility of this remote sensing method for inferring toxin concentrations from pigments. A sufficiently accurate image geolocation and projection was achieved, accurate within about 5 meters on average, to allow the selection of images overlapping the grab samples, accounting for any systematic errors or sort of offset errors associated with GPS and IMU measurements may further increase accuracy. NDVI applied to multispectral imagery over Lake Darling had a 43% correlation with chlorophyll A concentrations. This may improve with an increased image projection accuracy. Additional indices should be tested in future work, as many are sensitive to small changes in wavelength and bandwidth. The effect of sunglint as camera orientation and solar angle vary is substantial, and methods to reduce the propagation of such errors into index calculations to improve their performance should be investigated in future work. Cool. Sweet. Thank you.
That's great. We have just a couple minutes for questions.
Yeah.
And I think actually right now we just have a few. Okay, some have come in. First of all, gas vesicles strongly scatter light due to the difference in refractive index between the cell's cytoplasm and the gas-filled vesicles, in the same way that water bodies produce sun glint. If a bloom is localized at the air-water interface, are you able to delineate the contributions from gas vesicle-associated reflection versus water-associated reflection?
That's a really good question. I don't know much about that. I think sounds pretty interesting. But yeah, I haven't really looked much into that. We haven't thought about that too much, but thank you for that awesome question. I'll definitely look more into that.
Excellent.
I can assume that would have quite an effect, and it would be interesting to see if there's a way to pull that out of the reflectance data in some way, or at least account for it.
Okay. Is it difficult to fly on different days and register UAV positions, such as lat, long, height, in order to develop a time series of bloom states?
So as long as the weather is, decent, it's going to be okay. These are the way we did it was completely automated. So we pre-planned the flights and then just clicked go, and it went and it did its whole thing. So as long as the conditions, and there's regulations and sort of limits to how windy it can be, and what's going on in the atmosphere that prevents you from flying. But most of the time it's okay. And getting a license isn't that bad either. So if you're a lake manager and you want to do this, you have to do I think one test. It's an FAA license. So yeah, you can fly it pretty frequently.
Somebody's asking a little bit more specific information about the drone. What type size of drone is needed to fly this multispectral sensor?
So for this specific multispectral sensor, it's only about so big, and maybe maximum 15 centimeters long. And we used a DJI Inspire 1 drone, so a pretty small drone, and it had about 15 to 20 minutes of fly time, and we were able to do all of our flights with significant amount of battery left over, so we were safe with that. But yeah, the main thing is to this can be used for the areas of interest, or eventually at some point you can have this drone go and say, okay, well, maybe we should go here because this looks quite nasty, and then fly back, so you can kind of optimize how much battery you use. But yeah, small drone.
Okay, another drone question. Would a drone equipped with an RTK module be sufficient to mitigate the mosaicing issues over water where you might not have GCPs?
An RTK module. I'm not too sure what that is.
I'm not a drone expert, so I can't help you on that.
I'm not either.
Okay, well, we'll leave that one and we'll see what we can figure
That's
out.
interesting, Marv.
One final question. Nope. Maybe a couple more. We'll see what the time is. Do you have a specific protocol for grab samples? For example, subsurface integrate over 1 meter of column water column, etc.
Yeah, I think so. We just followed certain guidelines. It wasn't me exactly who actually collected the grab samples, but I think in general there was no scum. It was sort of homogenized, pretty homogenized throughout the water column, so we didn't end up getting any scum or actual visible algae cells in our samples. But I guess if there was scum there, you'd have to move it out the way and just make sure you don't get a bunch of gunk in there when you take a sample. But we did follow protocol, a specific protocol for collection. So we took them out and put them on ice and kept them out of light. And then that day, as soon as we were finished with all the collections, it went off to the lab. So.
Okay, one final question. Have you considered using UAV ground control points for interlacing?
So yes, and we kind of do use that a little bit. We have ground control points kind of that we on land. So that's why we look at those shoreline images, and we have, okay, well, we know, the location of this, so we can match them together. And then obviously once you get over water, there's errors associated with it, and there's no common landmarks between images, and you have all these changing reflections, between images. So the wind blows and you get waves, and as the drone changes, the landscape looks completely different from one image to the next, that you really can't match those images together or snap them together and like mosaicing those and making so one coherent scene is the main challenge here.
So I know that we're over time a little bit, but since we had one canceled presentation, you only have 2 more questions, and the other 2 presenters have been gracious enough to go back and type in answers. So I'm just gonna finish you out. How about that?
Perfect.
Okay, do sensors need a relatively high spectral resolution to get good results from the analysis you ran?
Yes, it depends. So for example, let's say you're looking for phycocyanin, that phycocyanin pigment has a distinct absorption characteristic at a wavelength that is not measured by our camera. So that is unfortunate. If you have a hyperspectral camera that has a very, very high spectral resolution, you can actually pull out more information and even distinguish between what's just, normal chlorophyll, maybe, I guess phycocyanin is more specific to cyanobacteria. So in our case, we're looking just biomass, and we're seeing if for this specific lake, for Lake Darling, can we relate biomass or chlorophyll to microcystin or toxins? Whereas if you want to have like a way more universal algorithm, it helps a lot if you have a very, very high spectral resolution, because you can pull out those in between the bands from a multispectral. There's so much information in there that you can use in your algorithm to increase your performance.
And your last question is totally related to that one. Basically asking, was it not possible to get a hyperspectral camera for the drone?
So the whole goal of this was to see if we could, match the performance of a hyperspectral camera for chlorophyll A specifically in small inland lakes. So we have that hyperspectral swath data, and in future work, we also have talked about maybe performing the same algorithms and seeing how well they compare. If we can get really close to hyperspectral performance, then that's great because lake managers don't need to go and buy an insane camera. They can just buy a pretty cheap multispectral camera and get essentially the same, if not very close results. But yeah,
Well,
having
thank you.
having a really nice big camera would be awesome, but that also the payload capacity, you'd need a bigger drone, so it just gets really expensive.
Of course.
So yeah.
Well, thank you for hanging around a little bit extra to answer all your questions. I appreciate that. I am going to share my screen one last time and thank everybody for attending this session today. Our next session will begin tomorrow morning at 8:30 Central Time using the same link that you used today. And our session tomorrow will focus on ecology and human health. Please take a moment to copy down these speaker emails if you have additional questions. Like I said, they've all been super gracious about answering questions above and beyond their time. So thank you to everyone who spoke today, even our extra Scott. We always have a few technical difficulties, but that was pretty smooth today. So thank you, everybody, and we hope you join us tomorrow morning.